So look, I'd like now to introduce Professor Ben Hayes, a professorial research fellow at the Centre for Animal Science at the University of Queensland. Ben has also joined the Queensland Alliance for Agriculture and Food Innovation. Ben has extensive research experience in genetic improvement of livestock, crop, pasture and aquaculture species, with a focus of integration of genomic information into breeding programs. Add to this, Ben has written more than 150 journal papers in this field. Ben led the successful development and implementation of a genomic breeding value for feed efficiency, which has been widely adopted by the Australian dairy industry. Ben was a co-inventor of genomic prediction, predicting the total genomic value using the genome-wide dense marker maps. This technology is used very widely in livestock and crops to predict future trait outcomes. The technology is also being used increasingly in human disease research. I give you Professor Ben Hayes to give an Australian perspective on genomics. Ben.
Thank you very much, Matt, and thank you to Graham and Eleanor for inviting me to be at this forum. It's always a great pleasure to come to New Zealand and interact with the scientists and the breeders, and I have to say I always learn quite a bit from coming across. So the microphone's not on? Maybe. Hold on, I'll just check. On the top. How's that? Excellent. Yep. Very good. So yeah, it's a great pleasure to be here. So I'm going to cover two topics in my course, in my talk. The first one has nothing to do with genomics, and the second one is all about genomics. So the first one is, across the Australian livestock industries, what are we breeding for? What are our breeding objectives? And the second topic is how are we going to get there as fast as possible. So what are we breeding for? As Andrew Cromie pointed out in his talk about beef largely, the real focus now is balanced breeding objectives. So the days of going hell for leather for a single trait like milk production, growth in sheep, those days are over, because we know if you do that, if you go for just one trait in your selection, it has bad consequences. There are traits which are unfavourably correlated, which will deteriorate if you just go very strongly for one trait. So just to show you what balance means across our different industries, the first one I'll touch on is dairy. And I'm sure most of you are aware that dairy got itself into quite a lot of trouble for going very hard for milk production for 30 odd years. And as a consequence of that, fertility really deteriorated. There's an unfavourable genetic correlation between those traits. So in the last decade or so, there's been more and more emphasis in the breeding goals on traits like fertility, to try and get a better balance. This is the three indices that are used in Australian dairy cattle breeding. I'll focus on the Balanced Performance Index, and I should also say that these were developed by Australian scientists together with Abacus Bio, so they played a key role in this. But the Balanced Performance Index, it includes about 50% of its weight on production, a fair chunk of weight on fertility, because that's a really important trait for profitability, mastitis resistance. Feed efficiency is a new trait that's in there now, so we have a breeding value for feed efficiency, and also traits like survival. So you can see a real balance across those traits. A bit closer to the industry you're in, lamb. So the emphasis has been on growth and also lean meat yield, using traits like fat depth and eye muscle area as proxies for getting to lean meat yield. Reproduction and fecal egg counts are also in there as well. Very recently, the sheep CRC, which is a very large research project that's underway in Australia, they've started developing indices and measuring the traits that are necessary to go into these indices with a focus on eating quality. And the motivation for this was really looking across industries and seeing the trouble that the pork industry in Australia and in other countries got into. In the pork industry, they went very hard for lean meat yield, and they in fact removed a lot of the intramuscular fat from the meat, and pork, the eating quality, the taste of it, went down quite severely. And in Australia, people almost stopped buying pork. The pig industry's turned it around, but the sheep CRC has taken a very forward-looking view, I guess, and said, we don't want to end up at that point. What we want to do is give our breeders the tools so they can maintain at least, if not improve, the eating quality of lamb. And you might think there is no problem with eating quality of lamb, but you just have to look at where the pork industry ended up to see that that could be a problem. And in fact, there are unfavourable correlations between lean meat yield and eating quality. So work that AGBU and particularly Andrew Swan has done recently is come up with an eating quality index that lamb breeders can now use to maintain the eating quality of lamb. And the traits that are the focus there, intramuscular fat is really the big one, but also tenderness through shear force. There's also quite a lot of taste panel data there. There's over 1,000 records of people eating lamb and scoring them for the taste. So that's a good development. Okay, beef. Beef, we have two industries. We have the Southern Australian industry, which is more about high quality product, and the Northern industry, which is about utilising pretty sparse pasture resources to turn off kilograms of live weight. The Southern industry, it started to look, well, it has been looking for a little while at meat quality as well as just growth. So intramuscular fat marbling is a big focus there. And there's starting to be a focus on feed efficiency as well. In the north, there has been a focus on growth, but it's been realised that reproduction, fertility, is really a very key driver of profitability of northern beef enterprises. So there's a lot of work going on to get good breeding values for fertility. And feed efficiency and meat quality is on the horizon, so beef producers can start to access those higher value markets. So across the three industries, it really is all about balance, and some good progress is being made. Now, how do we get there? As quickly as possible. And this is where genomics comes in. So for some of those traits like growth rate, you really don't need genomics. The traditional breeding values, because you can measure a lamb very early in its life or reasonably early for growth rate before you breed from that animal, you get a really good, accurate breeding value for growth rate. Some of the other traits, however, without genomics, you don't really have a hope of getting an accurate breeding value. And here I'm talking about traits like eating quality. So tenderness, intramuscular fat. It's very hard to measure that on extremely large numbers of animals. But what you can do is measure those traits in a reference or training population of up to 10,000 animals is what's happening in Australia now, and then get accurate genomic breeding values for those traits. And it's only when you have these accurate genomic breeding values that you're going to get a balanced selection direction. So if you have very accurate breeding values for growth, and although you might want to breed for intramuscular fat, if the accuracy of those breeding values is very low, you're going to make very little progress. But with the genomics, we have the potential to get accurate breeding values for these traits. Now, as other people have said before me, you really do need, or one of the key learnings of the past 5 years, is you do need pretty large populations of animals that have both the trait measured and a genotype before you can generate these accurate genomic breeding values. And the same is true for the two-step method, and it'll also be true for the single-step method as well, particularly for traits like eating quality, feed efficiency, and so on. So all the industries have faced this problem of how do you put together these large reference populations of animals that have genotypes and also have records for the traits. And you heard from Cromie how they tackled this in Ireland. They motivated the beef producers to record these traits by giving them some money and also showing them the benefits of recording what that would be. But I'll take you through how the Australian industries have dealt with this pretty significant problem. In dairy, dairy's a fantastic industry to work in because farmers, commercial dairy farmers, record lots of traits and they do a good job of it. So milk production, of course, but there's a proportion of the industry that also do a good job of recording traits like fertility, cell counts, and so on. So there's a lot of data there. So in dairy, basically all we had to do was trawl through the database, find which farmers were doing a really good job of recording traits like fertility, and then go in and genotype their cows. So over the past few years, we've genotyped 60,000 cows. This is Holsteins and Jerseys. And as a result of doing that 60,000 reference population, as well as bulls that have a lot of daughters are in there as well, the accuracy we're getting for our various traits is pretty good. So this is accuracy. The dairy people talk about reliability, but I've converted it to accuracies here. So for our balanced performance index, it's up to 79%. For production, 87%. And then for the other traits, because they're less heritable and there's less recording for the traits, it goes down. So survival, how long a cow lasts in the herd, longevity, 69%, fertility 69%. And feed efficiency is the interesting one. Feed efficiency is only measured on research farms. So it's cohorts of animals going through that get genotyped, have feed efficiency measured. The accuracy is much lower at 54%. But before we had the genomic information, that accuracy was closer to 0. Okay, and to emphasise the fact that when you have these accuracies that are reasonably high for these traits which have previously been hard to make progress in, I'll show you an example not from the Australian dairy industry, but from the US dairy industry. And I said that for dairy industries around the world, as a result of selection, very hard selection for high milk production, fertility declined. There's been a lot of effort to turn that around. But this slide shows the impact of genomics. So what you have is year of birth along the x-axis. This is when genomics came in here in the US, 2008, so it's only been with us for 10 years. On the y-axis is the daughter pregnancy rate. So as you go further up here, the bulls have the genetics to breed daughters with better and better fertility. And there's 4 lines on there, but I'll get you just to focus on the green one there, which is sires to breed bulls. The equivalent would be rams to breed rams. So the real pointy end of the industry, those really elite rams that you use to breed the next generation of rams. And if you look at what happened in 2008, there's this pretty major increase in the rate of gain for daughter pregnancy rate. And that, yep.
From 2000 to 2008, you were talking about a massive gain on the green line. And from 2010 to 2015, there's only a small gain.
So this is, in 2000, they put fertility into their index. Before that, there was no emphasis on fertility. So that's where these gains are coming. But what you've got to look at is the steepness of these lines here to see them going up. So after 2008, the line, well, there's a bit of noise around there, but it increases in its steepness. And the point of putting this up is to show that with the genomics, they're starting to make gains for this trait that they were making gains in, but it's certainly increasing. And if you talk to dairy farmers, they are starting to notice that slowly, that there are bulls out there that will have daughters that are more fertile. Okay, so that's the situation in dairy. It gets more challenging when you move to the other industries like lamb and beef. So the question here is, how do you put together these large reference populations that you can use to drive accurate genomic selection? So in Australia, we were very lucky that the federal government stepped up together with the industry to fund what is called the information nucleus. And this is basically using industry rams from studs around the country across a commercial U-base on various research stations, again, around the country. And the offspring from these matings get measured for a whole bunch of carcass traits and also meat quality as well. There have been about 30,000, I think it's a bit under 30,000, but approaching, animals genotyped, and that's resulted in genomic breeding values for lean meat yield, dressing percentage, as well as traits like intermuscular fat and shear force. So some of the top breeders are starting to use those genomic breeding values. And just recently, the team at AGBU, which deals with the genetic evaluations for sheep, has got their multi-trait single-step analysis up and running. Okay, beef. Beef's been more challenging because we haven't had this large federal government grant in recent history to fund these reference populations. So it's largely been about industry and the people that control the levy dollars coming together to set up some of these programs. So there's been the Beef Information Nucleus projects where steers are in a common environment and they go off to get slaughter data and meat quality data. Reproduction's interesting. There are 2 projects underway. One is really about measuring reproduction on research stations where you can get very good quality fertility phenotypes, like age at puberty, for example, which is a pretty tough thing to measure. There's also a project that I'm involved in that's starting to look at measuring these traits on commercial properties, similar to the Irish model. And again, in some breeds, this information is being used in a single-step analysis. And just to give you a bit of contrast between the dairy situation, days to calving is the fertility trait in beef cattle, and the accuracies are around 0.3 to 0.5. So much lower, still useful, but much lower, reflecting the fact there's not these very large genomic reference populations yet. Okay, so now looking into the future, what's in front of us? What do we still have to do? And there are two really big challenges, I think. One is, how do we maintain these reference populations or training populations for genomic selection? The challenging news, I guess, is we're always going to need them. We're always going to need big groups of animals that have genotypes and the traits that we're interested in, because we're going to have to update these genomic predictions or update the runs in the single step reasonably often. So that's a big challenge, and it has to be, well, it's going to be a mix of industry coming together with funding agencies to work out the best approach there. And I've been through 3 different approaches, and you saw from Andrew Cromie this morning how they're dealing with it in Ireland. The big challenge would be if the government dollars dried up, what would we do? But that's something that's luckily a little bit in the future. The other big challenge is how to make genomic breeding values work across breeds. So works particularly well in Holsteins, black and white dairy cattle, because you can get enormous number of records, you're dealing with a single breed, you can genotype lots of animals. So that's almost the ideal case. But in beef and sheep, we have multiple breeds, we have crossbreds, and we have composites as well. So how do we make these genomic breeding values work well across the breeds? And that's a big challenge that we have. So how to get there? This is our approach that we're taking. The 50K chip that's being used as the basis for evaluation in most industries for genomics around the world. It's fantastic if you're dealing in a single breed, Holsteins or Angus. Doesn't work at all really across breeds, or very difficult to make it work across breeds. So that means you need data from, or you need significant chunks of data from all the breeds you want to evaluate. There's a question about composites. I don't know much about the sheep composites that are used in New Zealand, but in the composites that are used in the beef industry in Australia, it really doesn't work particularly well at all. There's also this problem we have that the accuracy of the genomic predictions, it erodes quite quickly with increasing generations. So if you've got your reference population here where you've got animals measured and genotyped, you can go a couple of generations in front of that and you're okay, the accuracy holds up. But if you start going too many generations, the accuracy starts dropping off a cliff. And that's a bit of an issue because in dairy, and some sheep breeders are doing this as well, there's increasing use of technologies like IVF, breeding and using that to breed from young animals and to turn multiple generations over very, very quickly. And genomics works very well in those schemes, or should work very well in those schemes, because you can get the genomic breeding value to get accurate selections of which animals to put into the IVF schemes. But if the accuracy erodes over generations, you've got a problem. So what's the answer to this? We don't really know, but the approach we're taking is to move away from using a 50K chip to which, and the 50K chip, what it does is it relies on having markers close to the genes that are affecting your traits you're interested in. So it works by having, if you think of the chromosome, you've got a marker here You've got a gene for intramuscular fat here, and you hope that marker tracks that gene over generations. And that works well within a breed for 1 or 2 generations, but it doesn't work well across breeds or across generations. So what we're looking at now is sequence data. So rather than use 50,000 DNA markers across the genome, just read off the entire DNA sequence of the animals. So 3 billion ACTs and Gs. It's a lot of data, but somewhere buried in there are the mutations that are really affecting the trait, really driving differences in intramuscular fat, fertility, and so on. That's the approach we're taking. What you need to make this work is a large collection of sequence data. In cattle, this is well underway. We have what's called the 1,000 Bull Genomes Project, where we now have 2,703 bulls and a few cows with their entire genomes read off. And I have to say, this is a great example of international collaboration. There's over 30 different groups from around the world that are all contributing data into this common database, including New Zealand through Ag Research. And it covers a whole bunch of different breeds. Sheep are catching up on this as well. There's the Sheep Genomes Database, which is a collaboration between, largely a collaboration between Australia, New Zealand, and the US. And they've already got up to 930 genomes that have had this entire sequence read off. And when they line up those sequences, so you can imagine the sequence from one ram, sequence from the next ram, and so on, when they line them up and look for points of difference, they find 50 million spots that are different. And buried somewhere in there are the genes, the mutations affecting things like fertility, quality traits, and so on. So some of the people contributing to this are in the room, like Shannon over there and John McEwen. Okay, so that's the work we have in front of us, is to trawl through that data and find those mutations. If we can do that, and if the research is successful, those SNP variants, those sequence variants that really do explain differences between animals, that'll just go straight onto the SNP chips that are already being used for evaluations. So those will be extended to include those extra variants. And then that can be incorporated in single step and so on. Just to show you a hint that this might work, might be successful, I'll take you through an experiment we ran in cattle a couple of years ago. So this is about moving from the 50K SNP chip to using this sequence information. So what we did, we took, this is dairy cattle, we took the 2 breeds that we had a lot of information on, which were Holsteins and Jerseys, and together we had a reference population of about 20,000 animals that had really good fertility phenotypes, production phenotypes, and so on. We have a 3rd breed in Australia, which is patriotically called the Aussie Red, which is largely Scandinavian red in origin. They have much smaller numbers in the population, but they'd really like genomic breeding values as well. But they've struggled to get a big enough reference population to get accurate genomic breeding values. So the question for them was, could we use these other breeds to predict the Aussie Reds and get genomic breeding values for them? So all these individuals were genotyped for 50K SNP chips. We imputed that up to 600,000 high-density SNP genotypes, and then up to sequence genotypes, so the full sequence genotypes. The question was, how well could we predict with the 50K versus the high density genotypes versus the sequence. And we did this for two traits. One was fertility and one was temperament. And what you see there, the numbers you see there, are the correlations between the genomic breeding value and actual performance. So for fertility, it's actual fertility of the animals. And for temperament, it's a correlation between the predicted genomic breeding value for temperament and the actual temperament of the animals. So for 50K, if you're predicting into a third breed, you basically don't get anything at all for fertility or for temperament. The correlations there are pretty low. For the high density SNP chips, so 630,000 SNPs, you do a lot better. So you more than triple your accuracy there. Goes up to 0.22, 0.24. And then if you add sequence data on top of that, you increase by another chunk for fertility. Now these accuracies might not look that impressive, but remember we're coming from a zero base. So to go from pretty close to 0 to 0.28, that's a big jump. And in practice, the way these fertility genomic breeding values will roll out for a breed like Aussie Reds, a smaller breed, is we'll combine these are crossbreed predictions with any information we have for these smaller breeds. Okay, that brings me to the summaries then. So to go back to the beginning, what are we breeding for? All industries, I would say, are now breeding for balanced breeding objectives. Balancing up production, fertility, disease resistance, meat quality, because that's what is driving profitability. You could actually argue, does meat quality drive profitability? Because our lamb producers don't get paid per se on meat quality, but if you take a whole industry perspective and you look some way into the future, it's about maintaining market share for our lamb industry. Okay, and feed efficiency is up there as well. Genomic breeding values, whether they're single step, two-step, whatever, it gives you the opportunity to balance up your selection direction. So to improve fertility at the same time as improving milk production. Improve eating quality while at the same time, or at least maintain eating quality in lamb at the same time as improving lean meat yield, because it just gives you more accuracy for the breeding values for those hard to measure traits. Major challenges in the future are building and maintaining these reference populations. And that's really an industry discussion. For those of you around tonight at dinner, it's a really important discussion to have. And I don't think anybody's got the answer, the definitive answer to that question. Making genomic selection work across breeds. We're getting hints that this sequence data will improve the situation, but there's more work to be done there. And as you've heard this morning, there's a lot of work going into single-step methods, which is fantastic, I think, really good. But looking forward, we have to think about how do we integrate things like sequence variants into the single step. And Dorian touched a bit on that in his talk. Lots of people to thank. I won't thank them individually, but the people at the 1000 Bull Genomes Consortium, as I said, 30 groups from around the world, and also people at AGBU and the group down at DPI Victoria that I worked very closely with for many years. So thank you.
Just while one of you's getting a question ready for Ben, can I just ask the previous speakers to come up on stage? And we've got about 5 or 6 minutes just for a quick panel with questions and answers, but we'll give the first question to Ben. So Dorian, Andrew, Cheryl Ann, and Michael. And there's a couple of roaming mics out there, so if we've got a question for Ben.
Yep, Ben, really interesting talk. Maybe you can have a go at this and some of the other genomicists up there. The cost of all these technologies, you genomicists always want more fancy tests, more markers, etc. Can you talk about how the world's changing in terms of the costs of all these things? And Dorian and Andrew might have a perspective to chip in on that as well.
Okay, so two things there. The genotyping companies are reducing the cost of testing over time. And the reason they're doing that is there's pressure from other genotyping technologies, like things like genotyping by sequencing, that may be lower cost. And then the genotyping companies that develop SNP chips, they think, oh, if we don't reduce our costs, we're going to be out of business. So there's a constant sort of battle going on, which does reduce costs over time. And for example, Illumina, that develop a lot of the SNP chips, they've just brought out a new technology that should knock about $10 off the cost of testing. I shouldn't say dollars because I always get into trouble, but I will. That's about the number. So you will see the cost going down. The other side of the equation is how much value can we give back to breeders from a single genotype test. And I think we have to work really hard on that. So if you send away an ear punch or hair sample, you shouldn't get back just the genomic breeding values. That's great. But if you can also add into that parentage, all your genetic defects, horn polled, you can also use the genomics. It's really, really good for mating schemes that reduce inbreeding. So if you're worried about inbreeding, genomics is a great tool to start to control that inbreeding. So what we have to do is take that genomics information and turn it into multiple tools that breeders can actually use.